Assalamualaikum. My name is Mayutundi and my roll number is 032. I am from the Department of BS Biotechnology and my topic for presentation is GCMS. First of all, what is spectroscopy? Spectroscopy refers to the study how radiated energy and matter interact. The energy is absorbed by matter, creating an excited state. Spectrometry is the application of spectroscopy so that there are quantifiable results that can be assessed. Spectroscopy is used in physical and analytical chemistry because atoms and molecules have unique spectra. Now, introduction to mass spectrometry. Mass spectrometry is also called mass spectroscopy. It is an analytical chemistry technique that helps identify the amount and type of chemicals present in a sample by measuring the mass to charge ratio and the abundance of gas phase ions. A mass spectrum is a plot of the ion signal as a function of the mass to charge ratio. This technique basically studies the effect of ionizing energy on molecules. It depends upon chemical reactions in the gas phase in which sample molecules are consumed during the formation of ionic and neutral species. Mass spectrometry is a powerful technique that can identify a wide variety of chemical compounds. It is used to determine particles' mass, the elemental composition of the sample, and the chemical structures of large molecules. Mass spectrometry is the field that deals with separation and analysis of substances according to the masses of the atoms and molecules of which the substance is composed. It can often be used to calculate the exact molecular weight of the sample components as well. The instruments used in such studies are called mass spectrometers and mass spectrographs, and they operate on the principle that moving ions may be deflected by electric and magnetic fields. The two instruments differ only in the way in which the sorted charged particles are detected. In the mass spectrometer, they are detected electrically, while in the mass spectrographs by photographic or other non-electrical means. The term spectroscope is used to include both kinds of devices. Since electrical detectors are now most commonly used, the field is typically referred to as mass spectrometry. History of mass spectrometry. The foundation of mass spectroscopy was laid in 1898 when William Wien, a German physicist, discovered that beam of charged particles could be deflected by a magnetic field. In more refined experiments that were carried out between 1907 and 1913, the British physicist J.J. Thomson, who had already discovered the electron and observed its deflection by an electric field, passed a beam of positively charged ions through a combined electrostatic and magnetic field. The two fields in Thomson's tube were situated so that the ions were deflected through small angles in two perpendicular directions. The net result was that the ions produced a series of parabolic curves on a photographic plate placed in their path. Each parabola corresponded to ions of a particular mass to charge ratio with the specific position of each ion dependent on its velocity. The length of the parabolic curves provided a measure of the range of ion energies contained in the beam. Later, in an attempt to estimate the relative abundances of the various ion species present, Thomson replaced the photographic plate with the metal sheet in which was cut a parabolic slit. By varying the magnetic field, he was able to scan through a mass spectrum and measure a current corresponding to each separated ion species. Thus, he may be credited with the concentration of the first mass spectrograph and the first mass spectrometer. The most noteworthy observation made with the parabola spectrographs was the spectrum of the rare gases present in the atmosphere. In addition to lines due to helium that have mass of 4, neon have a mass of 20, and argon that have a mass of 40. There was a line corresponding to an ion of mass 22 that could not be attributed to any non-gas. The existence of forms of the same elements with different masses had been suspected, since It had been found that many pairs of radioactive materials could not be separated by chemical means. The name isotope, from the Greek for same place, was suggested by the British chemist Frederick Soddy in 1913 for these different radioactive forms of the same chemical species, because they could be classified in the same place in the periodic table of the elements. The ion of mass 22 was in fact a very stable, heavy isotope of neon. Now, the basic principle of mass spectrometry. The principle of mass spectrometry was based on Newton's second law of motion and momentum. A mass spectrometer uses this property of matter to plot ions by way of varying masses on a mass spectrum. From the law, we infer how much mass is relevant to inertia and acceleration of the body. This principle is applied to the aspect where ions with different masses to charge ratio are deflected by different angles in an electric and magnetic field. The basic principle of mass spectrometry is to generate ions from either inorganic or organic compounds by any suitable method to separate these ions by their mass to charge ratio and to detect them qualitatively and quantitatively by their respective mass to charge ratio and abundance. In this technique, molecules are bombarded with a beam of energetic electrons. The molecules are then ionized and broken up into many fragments, some of which are positive ions. Each kind of ion has a particular ratio of mass to charge, that is mass to charge ratio value. For most ions, the charge is 1, and thus mass to charge ratio is simply the molecular mass of the ion. The ion pass through magnetic and electric fields to reach detector, where they are detected and signals are recorded to give a mass spectra. Working of mass spectrometry. In a typical procedure, a sample, which may be solid, liquid or gas, is ionized, for example, by bombarding it with electrons. This may cause some of the sample's molecules to break into charged fragments. These ions are then separated according to their mass to charge ratio, typically by accelerating them and subjecting them to an electric or magnetic field. Ions of the same mass to charge ratio will undergo the same amount of deflection. The ions are then detected by a mechanism that is capable of detecting charged particles, such as an electron multiplier. Results are displayed as spectra of the relative abundance of detected ions as a function of the mass to charge ratio. The atoms or molecules in the sample can be identified by correlating known masses. For example, an entire molecule to the identified masses are through a characteristic fragmentation pattern. Now, components of mass spectrometry. The instruments, the instrument consists of three major components, that are ion source, analyzer, and detector system. Ion source for producing gaseous ions from the substance being studied. Second one is analyzer, that is used for resolving the ions into their characteristics mass components according to their mass to charge ratio. And the last one is detector system, that is used for detecting the ions and record the relative abundance of each of the dissolved ionic species. In addition, a sample introduction system is necessary to admit the sample to be studied to the ion source while maintaining the high vacuum requirements, that is 10 to 6 and 10 to 8 millimeter of mercury of the technique. And a computer is required to control the instrument acquired and manipulate data and compare spectra to reference libraries. With all of the above components, a mass spectrometer should always perform the following procedures. First one is produces ions from the sample in the ionization source. Second one, separate these ions according to their mass to charge ratio in the mass analyzer. Third, eventually fragment the selected ions and analyze the fragments in a second analyzer. Fourth one, it detects the ions emerging from the last analyzer and measure their abundance with the detector. That converts the ion into their electrical signals. And the last one, it processes the signals from the detector that are transmitted to the computer and control the instruments using feedback. Analysis of biomolecules using mass spectrometry. Mass spectrometry is fast becoming an indispensable field for analyzing biomolecules. Till the 1970s, the only analytical technique which provided similar information were electrophoretic, chromatographic, or ultracentrifugation methods. The results were not absolute, as they were based on characteristics other than the molecular weight. Thus, the only possibility of knowing the exact molecular weight of a macromolecule remained its calculation based on its chemical structure. The development of desorption ionization methods based on the emission of pre-existing ions, such as plasma desorption, that is PD, fast atom bombardment, FAB, or laser desorption, that is LD, that allowed the application of mass spectrometry for analyzing complex biomolecules, analysis of glycans. Oligosaccharides are molecules formed by the association of several monosaccharides linked through glycosidic bonds. The determination of the complete structure of oligosaccharide is more complex than that of protein or oligonucleotides. It involves the determination of additional components as a sequence of isomeric nature of monosaccharide and the capacity to form linear or branch oligosaccharides. Knowing the structure of an oligosaccharide requires not only the determination of its monosaccharide sequence and its branching pattern, but also the isomer position and the anomeric configuration of each of its glycosidic bonds. Advances in glycobiology involve a comprehensive study of structure, biosynthesis, and biology of sugar and saccharides. Mass spectrometry is emerging as an enabling technology in the field of glycomics and glycobiology. Analysis of lipids. Lipids are made up of many classes of different molecules which are soluble in organic solvents. Lipidomics, a major part of metabolomics, constitutes the detailed analysis and global characterization, both spatial and temporal, of the structure and function of lipids, the lipidome, within a living system. Many new strategies advances in glycobiology involve a for mass spectrometry-based analysis of lipids have been developed. The most popular lipidomics methodologies involve electrospray ionization, that is ESI source, and triple quadruple analysis. Using mass spectrometry, it is possible to determine the molecular weight, elemental composition, and the position of the position and nature of substituent in the lipid structure. Analysis of protein and peptides. Proteins and peptides are linear polymers made up of combination of the 20 amino acids that are linked by peptide bonds. Proteins undergo several post-translational modifications, extending the range of their function via such modifications. The term proteomics refers to the analysis of complete protein content in a living system, including co- and post-translationally modified proteins and alternatively supplied variants. Mass spectrometry has now become a critical technique for almost all proteomics experiments. It allows precise determination of the molecular mass of peptides as well as their sequences. This information can very well be used for protein identification, de novo sequencing, and identification of post-translational modifications. Next, instrumentation. There are four stages in mass spectrometry which we need to consideration. These are: first one is ionization, second is acceleration, third is deflection, and the last one is detection. Ionization. Atoms are ionized by knocking one or more electron to give positive ion. In acceleration, ions are accelerated so that they have same kinetic energy. In deflection, Ions are deflected to magnetic field due to difference in their masses, and in detection, beams of ions that are passing through a mass analyzer is detected by detection. Instrumentation, sample inlet. Sample stored in large reservoir from which molecule reaches ionization chamber at low pressure in steady stream by a pinhole called molecular lake. Ionization. Atoms are ionized by knocking one or more electrons off to give positive ion by bombardment with a stream of electrons. Most of the positive ions formed will carry charge of positive one. The initial sample may be solid, liquid, or gas. The sample is vaporized into a gas and then ionized by the ion source, usually by losing an electron to become a cation. Even species that normally form anions do not usually form ions are converted to cations. For example, halogens like chlorine and noble gases like argon. The ionization chamber is kept in a vacuum so that ions are produced and progress through the instrument without running into molecules from air. Ionization is from electrons that are produced by heating up a coil until it releases electrons. The electron collide with the sample molecules, knocking off one or more electrons. Since it takes more energy to remove more than one electron, most cations produced in the ionization chamber carry a charge of positive one. A positive charge matter plate pushes the sample ion to the next part of the machine. Ionization can be achieved by electron ionization, EIMS, chemical ionization, CIMS, and the last one is desorption technique, FAD. Electron ionization. Beam of high energy electrons, that is 70 electron volt, is emitted from filament, which is preheated to several thousand degrees. High energy electrons strike stream of molecules to strip and electrons from molecules and form a cation. Repeller is positively charged plates that direct newly formed ions to the accelerating plate. Acceleration is provided with potential of 1 to 10 kilovolt. Focusing slits direct the beam. Ionization potential required 15 electron volt and for bond pH, and required potential is 3 electron volt to 10 electron volt. Here, filament. Beam of electron is emitted from filament. Repeller is positively charged. Ion volume have electron ionization source that is about 1 centimeter, in which the source is very open, which means that the conductance of gas out of it is very high. Here is the lens that is used for acceleration and focusing of the ions. Chemical ionization. It is a soft ionization technique. Sample molecules are combined with ionized reagent gas, which is present in the excess chamber sample. Due to collision, various molecules are ionized by different mechanisms like proton transfer, electron transfer, atomic formation, etc. Any readily available gas are highly volatile. Liquid can be used as CIMS ionized reagent gas. For example, methane, ammonia, isobutane, methanol. In case of methane, proton transfer takes place. Varying reagent in chemical ionization, mass spectrometry allows selectivity of degree of ionization and degree of ion fragmentation. Choice of gas should be closest to proton affinity of reagent gas with that of sample to ensure efficient ionization of the sample without excess of the fragmentation. Using reagent gas with proton affinity matched closely to that of sample will keep molecular ion peak intact and smaller number of fragments. And the last one is desorption technique. It allows analysis of large molecular weighted molecules. It includes FAB, that is fast atom bombardment method. Fast atom bombardment method is an ionization technique that is used in mass spectrometry, in which a beam of high energy atoms strike a surface to create ions. When a beam of high energy ion is used instead of atom, as in secondary ion mass spectrometry, the method is known as liquid secondary ion method mass spectrometry. LSIMS. Sample to be analyzed is dispersed in matrix and placed in the path of high energy neutral atoms. Xenon and argon neutral atoms are used in these techniques. Collision of neutral atoms take place with molecules of sample to form ions and are forwarded to accelerating plates finally to mass analysis. In SAB, it uses neutral atoms to ionize sample, which means both positive and negative ions are formed and could be analyzed. Used for sample weights up to 20,000. These molecules basically include polypeptides, nucleotides, and oligonucleotides, etc. Matrix used in this technique should be non-volatile and inert. Acceleration. In this stage, ions are accelerated so that they all have same kinetic energy. Positive ion pass through three slits with voltage in decreasing order. Middle slit carries intermediate and finals at 0 volts. Deflection. There are several types of mass analyzers. Ions are detected by magnetic field due to difference in their masses. The lighter mass, more they are deflected. It also depends upon the number of positive charge an ion is carrying. The more positive charge, more it will be deflected. There are several types of mass analyzers. A time of flight analyzer accelerates ions to the same potential and then determines how long is needed for them to hit the detector. If the particles all start with the same charge and velocity depends on the mass, with the lighter component reaching the detector first. Other type of detectors measure not only how much time it takes for a particles reach the detector, but how much it is deflected by an electrical magnetic field. And the last one is detection. A detector counts the number of ions at different deflections. The data is plotted as a graph or spectrum of different masses. Detector work by recording the induced charge or current caused by an ion striking a surface or passing by it. The beams of ion passing to the mass analyzer is detected by a detector on the basis of mass-to-charge ratio. When an ion hits the metal box, charge is neutralized by an electron jumping from metal onto the ion. Thank you. That's from all my students.